Welcome to In Plain English, a podcast where we discuss scientific research in terms that are accessible to everyone, not just the experts. I'm your host, Jamie Moffa. Before we get started with today's episode, a few reminders. You can download the paper for each episode at inplainenglishpod.org by clicking on the episodes link in the main menu. You can also listen to previous episodes there. We believe in open access science for all, so the papers we choose will always be free for you to download. If you have a question or comment about a previous article, you can submit it under the Continue the Conversation tab. In future episodes, we will begin by reading and responding to some of the questions and comments that you send in. If you are interested in being a guest or presenter for a future episode, you can click on the Become a Guest tab on the website. You can also reach out to us on Facebook or Twitter at Plain English Sci. That's PLAINENGLISHSCI. To listen to this podcast, you can find us on Google Podcasts, Spotify, SoundCloud, or wherever you listen to podcasts. With that out of the way, on to today's paper. On today's episode, we will be talking about cancer, specifically all of the stuff that surrounds cancer cells and helps them to grow, survive, and spread. Our expert presenting today's paper is Gloria Marino. Gloria, would you like to introduce yourself?
Sure. Thanks for having me, Jamie. I'm super excited. So my name is Gloria. I am starting my 5th year of my PhD program. I'm in the Biochemistry and Molecular Biology program at Johns Hopkins University. I study cancer, as is probably obvious. Specifically, I study melanoma, which is a type of skin cancer, and how the environment around melanoma, our skin, changes as we get older, and then how those changes as we get older contributes to differences in how old versus young people survive and adapt to having melanoma.
Great, thanks, Gloria. And joining Gloria for this discussion are our two guests for this episode, Teodora Stoica and Jacob Walker. Teodora, Jacob, would you each like to briefly introduce yourselves?
So my name's Teodora. I'm a postdoc researcher at University of Arizona, and I'm looking at how brain networks change as we age.
And my name is Jacob Walker. I am a political activist, organizer, and I have started a nonprofit, among many other things. And right now I'm studying to go to law school. And I'm here on the podcast to be the person that doesn't know a lot about the subject. So I'm excited to learn from the people that are here. I'm really excited to be here. Thanks for having me, Jamie.
Yeah, thanks everyone for being on this second ever episode. And without further ado, Gloria, would you like to take it away with an introduction of the paper we're going to be talking about?
Sure. So today we are doing a literature review as our paper. So a literature review is a little bit different than a traditional scientific paper in that normally a scientific paper is very data focused. So lots of graphs, lots of charts and pictures, whereas a literature review is kind of what it sounds like. So it's much more where somebody takes a topic, a scientific topic, and they will summarize sort of what we know about this topic. And in a literature review, they will cite many different data papers to just kind of give us an idea of a whole particular field or something even very specific. So in this case, we are talking about a paper that's near and dear to my heart. It is called Tumor Microenvironment Components, Allies of Cancer Progression. So we're really talking about the tumor microenvironment today, which is a really cool and important topic that doesn't get a lot of love in the cancer field, but really should, and is sort of changing how we treat cancer. The tumor microenvironment is kind of a good name because it's really sort of what it sounds like. It is the small environment that surrounds wherever a tumor is in our body. So as I sort of mentioned, I study melanoma, and the microenvironment of melanoma, since it's a skin cancer, is the skin. And then kind of similarly for lung cancer, we think about the environment being the lung and everything that's around it. And what's important is that these environments contribute to how cancer grows. I like to say that tumors don't exist in a vacuum. Tumors exist kind of in mesh. They exist in 3D. They exist in a specific place, in a physical place. And that place has a lot of different components and a lot of different things that kind of cause cancer to change, spread, adapt, all of these different things. And so just kind of like as a brief story time of kind of the history of how we got here, kind of back, way back when we think about how we study cancer, a lot of it is just based on the tumor itself, right? What does the tumor look like? What are the cancer cells doing and how do we stop it? And that is all very important. But if we kind of start to think about, we'll say the mid 1800s, 1858 to be exact, this very, very smart man named Rudolf Virchow started noticing that cancer would often pop up in places that had chronic inflammation. And chronic inflammation basically means anywhere where immune cells are a lot. So when we think of inflammation, we think of when we get like a cut and it gets all kind of red and inflamed. That's what we're talking about. They can be anywhere in your body. So he noticed, he was like, it seems that cancer pops up a lot here. Maybe, where cancer pops up, or maybe when the environment is inflamed, it might cause cancer. This was in the 1850s, so he didn't really know, but he kind of, planted the seeds. Then in 1889, my good friend, Stephen Paget, he invented or kind of coined what is sort of become the tumor microenvironment field. So he coined what he called the seed and soil hypothesis. So seed and soil is interesting. What he basically did is, I think he was an MD, and if he wasn't, he got a lot of cadavers somehow. So he did, he basically noticed that women who had breast cancer, like very, very, very frequently, upon their death, he saw that upon autopsy that these women had cancer in their ovaries. And he was like, this is very weird, because if we're thinking about tumors as just cancer cells, and cancer cells can move and spread basically indiscriminately, you would think that among these hundreds of women, we would see kind of a random pattern of where this cancer winds up, all these metastases. But no, like many of them, the majority of them were in the ovaries. So that's when he started thinking like, maybe the cancer seeds only grow in a specific soil, in a specific tissue. So there is something sort of inherent about the tissue and inherent about the cancer cells that have to work together to grow. And now today, we really kind of think of the tumor microenvironment as a really critical part of cancer. The last kind of historical thing I want to tell you is we're going to fast forward to 2011. So 2011, Hardy Weinberg and Hanahan, they published this landmark paper. It's got like a bazillion citations, one of the biggest in the field, and it was called The Hallmarks of Cancer. And they basically outlined all the things that have to happen in order for a cancer cell to be a cancer cell. And one of them is that it has to have these very specific and unorganized and detrimental interactions with its environment.
Thanks, Gloria. I was wondering if you can talk about this really fun term that I highlighted in my reading of replicative immortality.
So replicative immortality is this very, very sci-fi and scary thing about cancer cells, is that our cells, our normal human cells that are not cancerous, they can only divide a certain number of times. And that number is relatively low. It's, somewhere between 40 and 70, but it's pretty tightly regulated. And cell growth, if you kind of think about it, it doesn't happen that much in our bodies. The only time cells really need to grow is if you get a cut or something and you kind of need to fill in that empty space, or if you need to sort of make a bunch of immune cells if you're sick. But other than that, our cells are just kind of at stasis, and they sort of grow whenever they need to fill in. So because of that, we have this super strict limit on our cells. The cancer cells bypass that and override it. So basically what they do is they just keep dividing and dividing and dividing forever. They are immortal cells. They never stop dividing.
So why can't we take this concept and live forever then?
This is a great question, Teodora. Many before you have thought of this, but you're right. No, you bring up a great point. Like, if our cells are capable of dividing forever, why don't we just do that then, right? The problem is that the reason that our cells don't divide forever normally is because when our cells divide, when they copy the DNA, there are errors, and the ends of your DNA don't get copied all the way every time. It's not very much, but there's kind of like a slight mismatch where it doesn't quite make it to the end. So you can imagine if you're not replicating your entire genome every time, and it starts getting shorter and shorter and shorter and shorter, it can start to get really bad really quickly. So our cells, luckily, have a fail-safe mechanism in place, and they have these things called telomeres, and they're basically like caps kind of on the ends of our DNA, and they're just nonsense. Like, it's just a bunch of genomic bases. They don't mean anything. They're never transcribed, never used. And so we have these caps.
Junk. They're junk.
Yes, we have caps of junk, but it's good because that way, every single time when the cell divides and it gets shorter and shorter, we're just losing junk and not anything important. So our cells know to stop dividing when we're getting close to the end of the junk cap, basically. This is important because anytime a cell starts to divide more than that, we are going to start to have problems very quickly. We're going to start having a bunch of different gene mutations. We're going to start losing gene function. And that is what happens in cancer cells. Cancer cells get mutated. They start losing, as they keep growing, they're losing all these crazy gene functions, and then they're upregulating stuff to kind of make up for it. So if we try and make ourselves live forever, we will just give ourselves cancer.
I'm really bummed, Gloria. That's all I got to tell you.
I know.
So as cool as replicative immortality is, or sounds, maybe not the best thing to have in
If you have cells in you that are replicating forever, you have cancer. Like, there is no cell in your body that, if it is dividing uncontrolled, is healthy.
Generally a negative thing if that happens.
Generally bad.
Perfect. Okay.
Well, we kind of think of cancer really as more of a disease classification as opposed to a specific thing. And so really, cancer, quote unquote, the disease, describes a disease where you have cells that are dividing out of control. And then that can be breast cancer. You have breast cancer. You have breast cells that are dividing. You have, melanoma cells. You have liver cells. Like, the type of cell, but like the disease overarching describes the phenomenon of having cells inside you that won't stop growing.
On that line of thinking, you mentioned the soil hypothesis. So how come each particular way that cancer grows in particular parts of the body is different?
It's a great question. It's kind of a combination of things. So you're starting with cells that are growing out of control, but they're not growing out of control the same way, as other people. So let's say person A and person B, they both come into the clinic, they both have brain cancer. That means that they both have a disease where some cell in their brain is dividing out of control. We have many different types of cells. So any one of those types of cells, or cells from that lineage, could be the ones that are growing out of control. So that alone makes it different. Let's say that person A and person B, it's the same cell type that's growing out of control. There are a lot of different ways that a cell can lose its regulation in growing. And we usually, like in the field, a common way to talk about it is sort of the gas and brake. So you basically have genes that when they are on, they act like gas. So genes get turned on and they start the growth process. And then you also have brakes. So you have genes that get turned on that stop growth. So you kind of have these start and stop genes. So you can get cancer one of two ways. You can either get a mutation in a gas gene where it's always on. So your foot is on the gas all the time. So no matter how much you try and hit the brakes, you are always going to be growing. Alternatively, you could also have a mutation in a gene that's the brakes, and then the brakes don't work anymore. So even though you're only putting your foot on the gas, as much as you normally would, without brakes, you're still growing. And there are multiple different genes that are gas and brakes, and you could have a mutation in any of those. And so the odds of person A and person B having the same mutation in the same gene, very low. And then you want to add to that, the specific microenvironment of their brain, this any sort of specific immune cells they have, it's very, 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 very different. So that's kind of why these things tend to manifest dramatically differently within people, even with the same quote unquote type of cancer.
So as I was reading this, I mean, I'm coming from a very different background from Teodora or Gloria or Jamie. So like, I'm kind of like zooming out a little bit and understanding a decent amount of what's in here, but also kind of like looking at what does this mean in like the larger context. And one of the things that jumps out to me really clearly is just the way we talk about cancer, especially in like the nonprofit space, which I have worked in organizations that have worked along like organizations like the American Cancer Society. And it's been in like healthcare advocacy capacities and stuff like that to like expand access to healthcare. And what is really interesting to me is I think there's a huge disconnect after talking to you, Gloria, and hearing like the initial start of this and reading this article about how people think about cancer and what the reality actually is. Because I think that there are a lot of people out there that are waiting for a cure to cancer, all capital letters. So I think that that's kind of an interesting thing that jumped out to me. So I'm wondering like just what your take is on that, because you have to see the fundraising that goes on with the American Cancer Society, and you have to see like, you have to notice even with talking with patients, the disconnect between like what the science is showing that these microenvironments are so important and that they're very different, the different cancers are very, very different versus what people's like the popular understanding of cancer. I'm just wondering what you think about that.
No, it's a great point, and you're totally right. I mean, I think sort of going back to the initial war on cancer, mid-century, sort of the idea of that initial funding for the war on cancer was that we're going to find the magic bullet that is going to cure all cancer, right? No matter where it is, no matter who you are, all cancer. And sort of as you guys have seen, even just reading this article or us talking about it, right? Like even the same type of cancer can be dramatically different between people, right? So the cure for cancer with a capital C doesn't exist because there just isn't a one size fits all approach to this.
I really think people would be so surprised to hear that. I mean, I think that that is very shocking.
Yeah.
You're totally right, Jacob. Like, I think that if the magic bullet existed, we probably would have found it by now, especially with, I think that people also get disappointed because they watch all this funding go into cancer research year after year after year, right? But I think really where we're going or where the future is, which we're sort of starting to see the beginnings of now, is personalized medicine for cancer. And I think a lot of different fields are sort of thinking about personalized medicine, but with cancer specifically, because as we discussed, it's just so different depending on who the person is. You really have to treat it person by person. And we're sort of starting to see the beginnings of that because tumor sequencing is happening now, right? Like, we've heard of triple negative breast cancer, right? That triple negative basically refers to what receptors or what things are sticking off of cancer cells that we could possibly target. And the way that we figure that out is we basically take the tumor, we crush up a biopsy of it, and then we sequence the cells and we figure out where the mutations are. And this is the case for a lot of different cancers because some cancers have common mutations. So not all cancers, but for example, in melanoma, there is a gene called BRAF, and it's a growth gene. And something like 60 to 70% of people with melanoma have a mutation in this particular gene. So we've been able to find a specific drug that targets that gene and basically puts the brakes back on. And that has helped numerous people, right? But not everyone. Like you need to know if you have that mutation in order to see if this medicine is going to work for you. So we're kind of, I think I see us sort of at the very beginning, sort of like the infant part of personalized medicine with things like that. But I think going forward, once we get more sophisticated with the amount of drugs that we have and the amount of sequencing we're able to do, I think you'll probably be able to get like, this is your tumor, person A. These are all your mutations, and we're going to target this and this. And then if and when we see it recur, we're going to resequence it, and then we're going to target, boom, boom, we're going to target the new mutations.
So back to my original question. If you do all of that, can you live forever?
You would probably live a lot longer, but I think that something else will get you like.
No, I'm genuinely interested in the science, but I've heard so much about how, for example, CRISPR can, and you can talk about more about what that is, Gloria, but I'm sure that has to do with an advent of personalized medicine in cancer treatment. But at the same time, if you take all of what CRISPR can do, all right, so we're going to edit our own genome, we can at least get rid of, not get rid of, but I guess make sure that cancer isn't one of the things that makes us die.
As far as CRISPR is a really interesting technology. So for, any listeners so Jacob, what do you know about CRISPR? Not to put you on the spot, but just to get a pulse on this.
I know of it as the thing that you put your vegetables into in the refrigerator and forget about them, or they freeze. So then, related to health, because they make you not eat vegetables, I think. So I think it's a net negative for me in my life. CRISPRs have been a net negative. So.
Okay. So CRISPR, it's a super interesting tech, but essentially it is a way of doing precision gene editing. So you can basically like put this system, if you will, into the cell, and then you can essentially like almost like laser target it to a specific gene, and you can cut that gene out of the DNA. Or they're kind of working on, changing, even just a single base pair, so you could change the idea that I think Teodora is getting at is that if you have a mutation, it is usually in one base of a gene, right? That's causing it to become a new gene or causing it to be weird. And you could go in with CRISPR, whoop, cut that, replace it with the right thing, and you fix the mutation. So the problem is that CRISPR works really well if you're doing cells in a dish, because you basically just like put a whole bunch of these, CRISPR systems in, and they'll go into all the cells, and it's kind of a closed system. And, and then kind of once CRISPR is done, you can then like sort the cells based on who was successfully CRISPR'd and who wasn't. We like can't do that in a person, right? Like that system doesn't work. CRISPR would really only work if we're talking about a single cell. So we could probably do it as just a sperm and an egg that had initially.
So basically that was going to be my question, because it sounds like if you CRISPR one cell and then, or one person, but then you couldn't just like put it back in their body, because then they would just have one CRISPR cell and one cell that had been CRISPR'd and then a bunch of other ones that haven't.
Right, exactly. Like that would do nothing.
Because you would have to change each one individually, which I imagine would take some time.
How would you even do it, right? How would you make sure that you were, if we knew how to get all the cancer cells, we wouldn't need to CRISPR them because we could just pluck them all out, right? Like that won't work. Circling back though to I think what you guys are on the right track with like, can we take your cells and reprogram them and then put them back in you, right? That is sort of like what you guys are getting at. But kind of the closest thing that we have, A, to truly personalized medicine, and B, to a cancer cure is CAR T cells. Have you guys heard of these or know what they are? No. Okay. It's literally CAR-T. And basically, so T cells, they're a type of immune cell in your body. And basically their function is that they kill cells that are infected with virus. So, kind of like now, if we were to get COVID.
Oh, because that's when they measure people with HIV. That's like one of the, isn't that one of the diagnostics that they do to determine something about?
You're right. So like in HIV, the virus attacks your white blood cells, and T cells and B cells are two subtypes of those so-called white blood cells. So yes, if you have low T cells, it basically means, hey, the virus is attacking your T cells, your HIV is progressing. So anyway, your T cells, your B cells are the ones that make antibodies. Your T cells are kind of the search and kill machines. So like essentially when a healthy cell in your body is sick, is infected with virus, it will basically throw up a flag that's like, help me. An antibody recognizes that and binds to the cell. That antibody is basically like a big giant X on the cell that's like, kill me, like kill this cell. This is disgusting. Get this out of here. And so T cells are the ones that do that. So given that sort of setup, you can imagine that T cells would be very, very, very helpful in cancer. If we could just get a T cell to recognize a cancer cell, we could make the T cells in your own body kill the cancer cells. Turns out they were able to do that. So CAR T cells,
Great.
I know, it's actually very happy. So CAR T cells, they take this actually, the downside is that this only seems to work in liquid tumors, so blood tumors like lymphoma, leukemia, lymphoma, all those different ones. But they take your T cells out and they reprogram them so that they recognize the cancer cells in your blood. And then the T cells, when they put them back in, the T cells will seek and destroy all those cancer cells. And it is remarkably successful. There's like one of the best like examples that they always use. There was a 10-year-old girl. Unfortunately, blood cancers like are predominantly found in children, especially leukemia. It's a very like,
That's sad.
it is. It's like it is mostly a pediatric cancer. It's very rare for adults to get it. So it was a 10-year-old girl. She had leukemia. She's like on death's door. CAR T cells are an experimental treatment, but she qualifies. They do it. That was 10 years ago. She is like right as rain, like has been in remission for 10 years. All of her scans come back clean every single time.
So since those cells are still in her body, then does that mean that if she were to perhaps have another occurrence of cancer, would the T cells just be like, nope, not today?
That is a great question. I actually don't know if we know the answer to that, because in your immune system, basically your body will create, antibodies and upregulate T cells and kind of get the whole immune system going when you're sick or when you have cancer or whatever. And then it dies down. And that's important, right? Like we don't want to have our immune system on high alert all the time. It doesn't really work like that. It's supposed to be kind of like crank it up, put it back down. So as far as the CAR T cells, what you really need in order for them to continue to be effective for years and years and years is you need what they call memory T cells. So they're sort of like, once your body is sort of like, okay, the danger is past, you're no longer sick with the flu, we can kind of die down again. It'll mostly just kind of let all these like T cells die off, like we don't really need them. But it'll keep like a very, very, very small few, kind of like in reserve. And so those we call the memory T cells. So they could, if need, they're kind of like our like reserve forces. So they could, if need be, straight back up. So we don't actually, I am not sure if we know whether CAR T cells are able to make memory T cells. Other thing that makes it complicated is if the cancer were to quote unquote come back, would it be exactly the same?
Okay, I have a quick question, a very, very selfish personal question. I live in Arizona. There's lots of sun. Sun is out, like I want to say 24 hours a day because it seems like that. What is the number SPF? What's the magic number that I should be wearing at all times on all parts of my body?
This is an excellent question. I'm a huge sunscreen advocate. A common thing that people think is that you can get a base tan and then you're fine, or if you are a person who is very rich in melanin, that you are fine. But the truth is everybody needs to wear sunscreen. I think most people kind of think that SPF is like a linear scale, like 50 gives you like 20 more protection than 30 or whatever, and that's not really the case. So You really need SPF 30 at a minimum. Anything beyond like 50, you're not getting like that much more coverage.
Wait, what's wrong with a tan?
Oh, well, like, no, don't tell my PI this, but I also love to be tan. I love to go to the beach. I always wear sunscreen, but I will still usually get tan even if I wear sunscreen. And the truth is that there's really no such thing as face tan, because if you are tanning, when we get tan, we get darker because there's more melanin in our skin. So melanin is what produces our skin pigment. The only people who don't have any melanin are people who have albinism, who are albino. Everybody else has some level of melanin, and that kind of depends on what your relative skin tone is. So melanin is produced by cells in your skin called melanocytes. So they produce melanin, and it is actually fascinating. So these melanocytes, in response to UV radiation specifically, in response to the sun, melanocytes will start to produce melanin. And basically, melanin looks under the microscope like these black masses, just like totally black. And you can see that the melanin will start to sort of cover your healthy cells, kind of like an umbrella. Like it literally is
fascinating.
Like, yeah, it's like the melanocytes like bloom almost, and it's like these big black shades that are trying to shade your cells so that they aren't getting as much radiation. When you are tan, your body is like, holy crap, I need to protect my cells. So I need to put up these, safety umbrellas. Once you've gotten to that point, there's already some kind of sufficient damage. Obviously, if you are wearing sunscreen, it is better than nothing, but avoiding the sun as best that you can is usually best.
So melanoma is when you have, so the melanocytes have cancer, or which part of you has cancer? The melanocytes?
Yes, the melanocytes are the like cell of origin. So the melanocyte starts to divide uncontrollably.
So we don't have any more umbrellas.
Or you have like too many umbrellas that aren't really doing their job. Another like common critique of sunscreen is that you aren't going to get enough vitamin D, because when you are exposed to the sun, you make vitamin D. But like if you could eat like one vegetable a week, you've probably got enough vitamin D.
Can we talk about the actual tumor microenvironments? So what are the components if you have like a melanoma in your skin? What is all the stuff around it, and how is that affecting how the cancer is growing and spreading?
Okay, this is a great question. So I really like studying melanoma for the tumor microenvironment because the skin is a really, really great subject to study. So sort of what I study and what I very, very biasly think is like one of the most important parts of the microenvironment is actually something called the extracellular matrix. So, or the ECM. So the extracellular matrix is again very aptly named. It's basically a whole bunch of different proteins that makes like the mesh that everything in your body sits in. So basically anything that isn't an organ or like a blood vessel is extracellular matrix. It's kind of like all the filler. So ECM, it has a lot of like important functions, including sort of retaining water and kind of allowing for structure. And so the water retention part is really important because as we get older and we get wrinkles, what's basically happening is your ECM is starting to break down. And it's not like through any really fault of your own, but like it just happens over time. But if we circle back to cancer and why the ECM matters in it, sort of what we see is that over the course of the natural aging process, we start to see this breakdown in the structure of the extracellular matrix. Interestingly, we see a very, very similar kind of breakdown in cancer. So when a tumor like forms, one of the first things it's going to start doing is it's going to start breaking down the ECM around it. And it does that for a lot of reasons. One, your ECM is like protective and providing structure, and your tumor doesn't like that. And it doesn't want that. It wants to be free. But also, your tumor wants to grow and it wants to move. So cancer cells that want to move, that want to get out of the primary tumor and kind of travel throughout your body, if they try and go through the ECM, it's going to be really difficult because it's just like squishy, meshy, it's really kind of hard to get through. It's designed that way. So what cancer will start to do is it'll start to send out a whole bunch of little proteins and a whole bunch of little, things into the ECM that will break down the structure and will make it stiffer. So when the ECM stiffens, the cancer cells can start to use it more like a road. So rather than it like cancer cells kind of walking in this like squishy swamp, it gets really stiff, and then they're easily able to kind of crawl along your ECM and move.
You know what, Gloria? Cancer.
It's horrific.
Yeah, it's horrific. And I think the name of this podcast, should be Cancer's a Bitch.
Yeah.
You are right. The like cool irony of cancer, right, is that it's your own cells. Like, it's not like it's an alien thing. Like, it's your own cells, and all they're trying to do is survive. One of the things that they need to do in order to survive is they need to start changing their environment to help them. So changing the ECM, huge step. And actually, some specific cancers, I think like some of the most common examples are breast and liver. They can actually, when they do initial biopsy, sometimes they will look at how stiff your ECM is when they do the biopsy, because that is a huge prognostic marker, meaning like if they take the biopsy and they see that you have a really stiff ECM, they can kind of start to adjust your treatment. They can kind of start to adjust maybe even your survival timeline, because they know stiff ECM means this cancer can spread a lot faster. It's a lot more aggressive, all of these things.
What are stages?
Stages of cancer. Great question. So generally speaking, if we're thinking about what we call solid tumors, so any tumor that's not in the blood, it basically refers to how quote unquote deep the tumor is, if that makes sense. So if you have a melanoma tumor, it usually starts as a weird looking mole, and it's just kind of on the surface of your skin. So that's usually stage 0 or stage 1, however you want to think about it. And eventually, your skin cancer, instead of sort of growing out on your skin to be larger, it will start growing into your skin. It'll start growing vertically. So once it starts growing in, then we start to hit all of the stuff in the microenvironment that we don't want it to be near. Then that's when we start to get into the ECM, when we kind of breach that skin layer, if you will. The level of breach determines the staging. So usually once it's kind of breached the skin and it's deep, then it's a stage 2. Stage 3 is usually what they'll do is if they think that you have a aggressive melanoma, or they think it's stage 2 or higher, they'll do a biopsy of your lymph node, whatever the closest one is. And so if they see cancer cells in your lymph node, then you get stage 3, because that means that they've already traveled, right? Stage 4 is usually reserved for anything that is metastatic. So metastatic means that you have a secondary tumor somewhere else in your body, anywhere else. So that is sort of the level of staging. So obviously, catching cancer really early is what is really critical to treatment right now. So if you can catch it at stage 0, 1, even 2, before it's really started to travel, that's usually when surgery is really effective, because you can cut it out and that things haven't moved that much. Once we start getting into stage 3 and stage 4, when things are really moving, we're talking about trying to track cancer cells through your body, and we're just not there yet medically, right?
So something that I thought was pretty interesting when reading the article, also with the talking about the microenvironments of like where a tumor happens to be, if it happens to be, near a bony place in your body, or near like kind of a fatty place in your body, or wherever it may be, is kind of like, I kind of thought about the local like news stations that are always like, oh, red wine is like good for like not getting cancer. And then like a couple of weeks later, they'll be like, oh, it's like red wine is really bad. It can give you cancer, or it can be bad for you. They have these like constantly conflicting things. But then reading this and then hearing about how different all of the different cancers are, I'm like, well, obviously, like, for one type or one part of your body, like something could be good and contain something that's good. And then for another part of your body, if something else or a different type of cancer happened, like it could be really bad, or like, oh, this is something people complain about all the time. Like you can't really get good health advice and the local news just throws it on there with no context. And then I'm just like, well, no wonder, because everyone is mistaken about cancer, like that, the very base fact about cancer. Like,
you are exactly right. And it kind of ties into like what Teodora was talking about earlier too, is that like, that's sort of the reason we don't have this magic bullet, right? Like every organ, if we even think of cancers as like organ specific, right? Every organ needs different things, right? So it's not like we can treat lung cancer the same as we treat brain cancer, the same as we treat like a sarcoma that comes from the muscle, or like a heart, or like all of these things are so dramatically different that exactly, like when you start talking about anti-cancer, again, it's like, what cancer are you talking about?
I will also say, Jacob, that whenever they say that red wine is bad, I immediately turn everything off. I'm like, no, you will not tell me red wine is bad. You will not tell me that, like.
Well, what's interesting, kind of thinking about like sort of cancer prevention or whatever, it is untrue that eating blueberries is going to like prevent you from getting cancer, right? Like what is interesting is that I don't think last year, because obviously COVID really messed with everything, but I think in 2019, it was one of the first years that we actually had a decline in global cancer rates. It wasn't much. It was like somewhere between 1 or 2%, but it was still a decline. And like, especially like if you're looking across the globe, it was a pretty big deal. Interestingly, like it wasn't necessarily because we have some new great cutting edge, treatments. It was really that global smoking rates are the lowest they have been in a very long time. And so lung cancer, like far and away, is like killing people at a massive rate globally. And it's mostly due to the fact that so many people around the world smoke. And so, there are so many cancers that you really can't prevent. Like, these cancers that just arise from random mutations, it's just bad luck. Like there's no amount of blueberries or kombucha or green tea that can prevent that. But there are cancers that are definitely preventable, or at the very least, like you can massively reduce your chance of getting it. And lung cancer, liver cancer, and melanoma are these big three. And lung cancer is obviously, not smoking. Melanoma is avoiding the sun, wearing SPF, and then liver cancer is not being an alcoholic, essentially, not like over consuming alcohol. Unfortunately, the things that we know work, like not smoking and not tanning, people don't really want to stop doing. So let me talk a little bit about sort of the role of blood vessels, kind of when we think about the tumor microenvironment, because this is another really sort of important part, both blood vessels and lymphatic vessels. So your blood vessels and your lymphatic vessels, blood obviously carrying blood, lymphatics is essentially carrying all of your immune cells and also cell waste. Both of these vessels are kind of all permeated throughout your body, and they basically kind of weave through the extracellular matrix as well. So again, if we're kind of going back to skin, if we're thinking about the surface area that we can all see that isn't very deep, where stage 1 cancer might occur, and we start thinking about stage 2, that would be if it goes down into that extracellular matrix where all these vessels are kind of living. And so again, if we're thinking about cancer, and cancer is our own cells, our cells need nutrients and oxygen to survive. What cancer tumors and cells will start doing is they will start sending out signals that are like, hey, I need more nutrients. I need more oxygen. I need to like be fed, essentially. And your body will start to create more blood vessels that will go and feed the tumor. That's sort of another thing that when physicians or whatever will look at a tumor, they will look at its quote unquote vascularization. How many vessels are in it, right? Because again, if it's got a lot of blood vessels connected to it, it is aggressive. It is going to get big. And the other
Hungry tumor.
Yes, it's very hungry. And that's the thing, is it kind of starts to like siphon, you know what I mean? Like your blood, like your body's just pumping blood for everybody, and then your tumor is just like siphoning it for itself, basically.
So, I mean, so if you have say you have a melanoma that has advanced and it's like on your arm and it's gone down and it's like gone to a different place, like what makes cancer actually fatal? Is that when a cancer cell lands in a place you really don't want it to land, that's like vital for vital for the continuation of life, and then it grows there to the point that it disrupts?
That is essentially what will eventually kill you, is that like...
Okay, so it goes back to the microenvironment also then of like the next place that it lands to, even at like the end stage of the whole thing.
Yes, because basically, like, you know, you could die from a primary tumor if it's in a really, really bad spot, right? But like really what kills you is that, and this kind of goes back to what we were talking about the blood vessels, is that the main way that cancer cells start to move is they enter your bloodstream, because your bloodstream goes everywhere. It's like a superhighway. So if your cancer cells can enter, zip into your bloodstream, they can go anywhere and they can just leave at any point. So the more blood vessels that a tumor has going to it, right, it's got like all of these highway entrances right there. So the odds that at least one cell is going to make it in is really high. And so really what tends to happen is, like you said, you know, let's say you have melanoma, one of the places it tends to metastasize to is the lung. So, you know, you're battling this melanoma, it's sort of growing in your skin, and then it gets to your lung. Okay, well, now you have this thing growing in your lung, and like, can you like get enough oxygen? Okay, even if we were to like get rid of one of your lungs, oh, shocker, like it's moved to your other lung. You know what I mean? Or like,
yeah,
cancer also tends, or melanoma at least, it also tends to metastasize to the brain. I mean, maybe we can remove it from your brain, but then it might get into your spinal column. You know, it's just like,
yeah,
it eventually just gets that you're chasing this thing around the body, and it's going to eventually get somewhere where it is just incompatible with life, basically.
I've never seen anyone in personally die of cancer like in front of me, but on the media, I see that they die in pain. Why is there pain?
So often there is pain because like you have something growing that is pressing into other like organs and pressing into space. You know what I mean? So it's pressing on nerves that are sort of all around it where things shouldn't be. It's like, yeah, like imagine the pain. It is, if you like Alien or like a parasite, like imagining the pain of something growing like in the center of your arm and growing, right? Like
it's see.
Just pushing. Yeah. So that is like usually where the pain comes from. If you want to get really dark, the most like intense pain I hear for cancer is bone cancer. And I think it's because it's like so deep in there, and it's like it is always going to push on things.
Right. Because there are some cancers that you don't know that you have. So you're not in pain. You don't even know.
Right. So one of the like biggest perpetrators of that is pancreatic cancer. Like of all the cancer types, so since 1990, your prognosis for every single cancer type has improved, some more significantly than others, but all are better, except for pancreatic cancer. We have made no progress on that tumor since whatever, the dawn of time. And a lot of the reason is because your pancreas is this like smallish organ. It's kind of tucked up in there. So it's not like it would, you know, some people will see cancers pop up, like, you know, if they have something up with their lungs or if they have something up their bowel, like sometimes it'll just pop up, like they find it by accident. Like your pancreas is not really part of like a normal screen. And it's so sort of hidden that by the time you start to experience pain, the size, the amount that that tumor has to have grown is huge. And usually when a tumor is very big, some cells of it have already moved, have already gone off. So the problem is not necessarily that we don't know how to treat pancreatic cancer. The problem is that we can't detect it early enough. So most people who get it they're usually in stage like 3 or 4 already. And then by that point, you're already at the, oh crap, we have to chase this around the body stage. So that's why your prognosis is so bad. Not because that cancer is necessarily like intrinsically better or worse than another one, but it's just our ability to detect it is so bad.
I feel like cancer's like real estate almost. Like it's all about the location.
And it's a lot of like luck or bad luck. Like it's so random. I mean, really, because like they kind of call it like the 2-hit hypothesis in cancer, which is sort of like how cancer starts. Our body has a lot of fail-safes built in, right? We have lots of pairs of gas and brakes. We have lots of things that can compensate for each other should something go wrong. You know, we have a lot of fail-safes built in. So basically what they think happens is one of the fail-safes that your body is built in is that you have 2 copies of every gene, right? One from mom and one from dad. And the idea is that if one of those gene copies were to become non-functional for any reason, no worries, because you've got a whole second copy backed up. So you could just stop transcribing the crap one and, you know, we'll just go with the other one. So the 2-hit hypothesis is basically that lightning strikes twice and you get 2 random mutations in the same gene. So you get, boom, they knock out one copy of your gene, boom, it knocks out the other. Now you have a non-functional gene. And so kind of like stemming from that is BRCA mutation is a very like famous mutation in a specific gene called BRCA that makes women more susceptible to getting breast cancer. So if breast cancer runs in the family, women will often get screened for this mutation. And you have, it's something crazy. Like you have like a 40% higher chance of getting breast cancer than the average, some like crazy metric like that. And the reason is that if you have a mutation in this gene called BRCA, that means that one of your copies is already faulty. So that means that you are starting with only one functional copy.
So
right?
It's the 1-hit hypothesis.
And then it becomes the 1-hit hypothesis.
Yeah.
Exactly,
Okay.
right? So like then the odds are not in your favor because like if something happens, you're screwed, right? So that's kind of where it comes from. And it just so happens that that gene is involved in DNA repair. So it fixes mutations. So if you have a non-functional gene that is supposed to fix gene mutations, it's bad news bears, right?
Interesting. And then I think when we were talking, you said that like just the process of aging just provides more opportunities for that firing to not be good. Basically.
Pretty much. I mean, essentially, you know, kind of how we talked about like with, you know, telomeres are like big junk caps on our DNA. DNA transcription is 99.999 or whatever % accurate, but it is still 0.001% inaccurate. So we have all these fail safes in place. We have DNA repair mechanisms where, you know, we kind of do like when cells are dividing, we do a quick check. So these proteins will go zoop along the DNA and they'll make sure, yes, everything's good. If they see something that's wrong, they'll fix it. With those in place, we're at 99.999, which is really, really good. But there's always a chance that something goes wrong. And, you know, if that goes wrong in, again, one copy of a gene in one cell, no big deal. 2 copies and that cell starts to go haywire, then it's trouble, right? The last like bit of this paper that I wanted to talk with you guys is the immune system and immune cells as members of the tumor microenvironment. And we kind of touched on this when we talked about like CAR T cells and stuff, because your immune system is built to get rid of threats in your body, whether that be viruses or parasites or bacteria or even cancer cells. And there's actually a lot of evidence to show that throughout your entire life, your immune system is very good at getting rid of potential cancer cells, like early in your life. Like there's evidence to show that like we have a cancer cell or two, or something that could become cancer floating around that our immune system is just taking care of. So it is actually very, very effective. But the problem is that once you get a cancer that really, really, really gets going, it kind of overwhelms your immune system. And you start to get this thing where they call it like immune exhaustion or even T cell exhaustion, where your immune system, like we talked about, we want to turn on and then we want to turn it off. Once your immune system is on and it's trying to get this tumor for, we'll say days and weeks and months, it just loses its effectiveness. Like the cells get tired, like they just don't work as well. And then that kind of translates to sort of the rest of your body, because your whole immune system is just not as functional as it should be, because it's just been on all the time. And so it really just kind of makes you a little bit more susceptible to other things. So when we kind of go back to thinking about like diet, for example, something that I can't remember who actually coined this phrase, but they basically called cancer the wound that doesn't heal. You have these cells that are growing, trying to fill in a missing gap, and you have your immune system that's up and active, trying to fix it, but then it just never heals. So you just are in this like chronic state of like, we need to fix this, we have something wrong. So if you have a very poor diet, or if you are very overweight, your immune system is already kind of high. You just have kind of a lot of toxic things coming through your body that your immune system has to deal with.
Is that coming from the food or from the impacts of being overweight, or both?
Both. So
okay.
If you kind of, there's some evidence to show that, like, if you're eating super processed food, it causes kind of inflammation or immune cells coming into like the gut to kind of calm everything down. And when you're overweight, also often your heart has to work harder, which does kind of impact like how your like aorta works, so that can become inflamed. As well as your joints, they tend to have a little bit more stress on them, so they can become inflamed. So you kind of have like all of your body's trying to kind of put out fires a lot with your immune system. So the reason that these things kind of sort of preclude you to cancer is, you can imagine, if our immune system is sort of like picking off these cancer cells periodically because it's like working properly, if your immune system is already like doing 800 bazillion other things, like a cancer cell might slip through the cracks. So that's sort of where the connection is.
Okay.
So it's not necessarily like that you are eating this one particular thing, or that you are not doing this one particular thing. It's that like you are creating this environment in your body where your immune system is like hyperactive all the time. And then, like, ironically, the hyperactivity leads to less effective immune system.
So if you don't get sick often and you have a really good immune system, then does that mean that so the healthier you are in general, like if you don't get colds and does that mean that also lowers your risk of cancer?
Yes. Like the thing about cancer, right, is that it's random. So you could have something in your DNA, that is just kind of a ticking time bomb. But it's one of those things where it's like your eating habits, your exercise habits will not contribute to increasing your odds of getting cancer, if that makes sense. So it's more of like, it's less of like, oh, you're less likely to get cancer than somebody else, which is probably true. But like, just because it's so random, like, it's hard to say for sure. But you are doing much more to decrease your chance of getting cancer.
It's more like a harm reduction than.
Yes, because cancer is random, right? There's like not very much you can control, but like what you can control, like of the things you can control, we know that being overall healthier, like making sure your immune system is sort of like able to function correctly, turn on and then turn off. All of these things kind of help you just be like, give yourself the best chance, basically, right? Like, I mean, I think like the good thing about, like, if we want to sort of end on a higher note, or at least like what is exciting, I think that personalized medicine is going to be the way of the future, and I'm excited about it because I think that cancer is really kind of paving the way for personalized medicine with a lot of other diseases. I think that, for example, like neurodegenerative diseases, you want to talk about the brain and how complicated that is. There are so many different things where I think that the medical field has really tried to simplify it, where I think that a lot of people could really benefit from personalized medicine in so many different, facets of care. So I think that kind of as cancer care becomes more personalized, my hope is that that will really extend to a lot of other things. So that maybe one day, like, way, way in the future, it's like, okay, we have like the Jacob like health plan. Like we, sequence your tumor. We know that you have like a non-functional copy of this gene that, may make you more likely to have heart disease. So we're going to make sure that we're like screening for that, like that I think is like my pie in the sky dream of healthcare. The other thing I'm excited about is actually the FDA approval of mRNA vaccines for coronavirus is a huge win for the cancer field because mRNA vaccines, as you guys might know, have been sort of in the works for the last 20 years. We really just needed apparently a global pandemic to really light the fire into the FDA's butt to get them going. Being able to just use a short mRNA sequence to program like essentially your immune system, it's kind of like CAR T cells, but in a shot. Because like essentially what you're doing is you are priming your immune system to recognize a certain threat and make antibodies against something. And because it's like a short mRNA sequence that they put in the vaccine, you can easily switch it up and it's very cheap. Like you just change, the sequence and then you just put it back in. So for cancer patients, that could be huge, right? Like rather than having to like take the T cells out and change them in a dish and put them back in you and, wait and do all like, you could just give somebody a vaccine to make your immune system recognize your cancer cells and then they'll wipe them out. And then if they pop up again, you can just give them a new vaccine. So that is like a super exciting. And again, it kind of ties into like personalized medicine. But I think those two things, at least of what we see now, I think will really kind of radicalize cancer care in a really good way.
That is hopeful, I think. And also hopeful that maybe we can get ahead of some of the variants with COVID as well.
Yeah, exactly. I mean, I think the other thing too about us making progress with cancer is a lot of the progress that we've made has been because we have lowered the ages of screening for a lot of cancers. We started screening earlier, and we have a lot more advanced technology for imaging, right? We have like MRI machines, we have CAT scans, we have all these different things rather than just, like an X-ray or whatever. So I think as well as our technology gets better and better and better, we can increase our resolution of imaging. And because it's a technology thing and it's just like exponential, I mean, I have no reason to believe that we couldn't one day achieve single cell or maybe multi-cell resolution in imaging. And that could be like really important. Like that could really radicalize, how early we're able to detect things.
But I wonder if there's like a proxy that we could get to that we don't have to put people in an imaging scanner, but I don't know, like have a really consistent way of saying, okay, we're going to take this piece of your blood and then figure out where your cancer is. I don't know. I just feel like there's so much testing and testing costs money and people don't have money to pay for the testing and testing money, time, etc. And there's just elongates this process of actually getting the freaking treatment. So if there would be like an easy way to detect it, that would be even better.
Yeah, I think a lot of, again, Teodora, she's on it with what people have thought of. So like what you're describing is what we call biomarkers in the field, but basically like similarly, can we take a sample of blood and figure out what type of cancer you have? Sometimes, yes, but if you have cancer, like if you have cancer cells in your blood, it is very, very, very bad news, right? So It's not incredibly helpful yet. But hopefully we can get there. They're kind of starting to look to see if they can find tumor DNA in the bloodstream, which doesn't like, the good thing about when cancer cells get into your blood is going at a very, very fast rate. So it's kind of rather, maybe not so much them getting onto a highway, but more like them going to like rapids. So a lot of times they don't survive in your bloodstream and they'll like blow apart. But there are traces of DNA that are left behind if the cancer cell gets blown apart, right? So that is something that they've tried to look at. But I agree, that would be obviously the best. You could just do a little finger prick.
Yeah.
But
conversely, come on, Gloria.
But like a good thing, like if we're talking about imaging and sort of if it's technology necessarily gets better and it gets cheaper. That's just like how it goes, right? I mean, obviously the most high tech, is going to be the most expensive, but like as we increase our resolution, our technology, what was considered state of the art is going to become less state of the art, but be cheaper. You know what I mean? So like once we're getting into like these insane imaging, all of a sudden maybe everyone doing an annual MRI isn't going to be that expensive.
Right.
Yeah. So, it's kind of one of those things that I think that could really work in people's favor, where, even if it's not the best resolution, it's way better than what we have now, and it can be cheap and relatively easy.
Yeah, and I think what's been interesting about this entire discussion is just how you really, if you like scratch the surface of all, it seems like on cancer research and the search for a cure or whatever you wanted to call it, or the whole ecosystem around solving this problem, which is really a lot of different problems that are just kind of tangentially related to each other. Mm-hmm. I think what's interesting to me is like the way you have to change your expectations. And I think like it's interesting, Gloria, to hear from you and hear how excited you are about technology and about earlier screening and stuff like that, because I think we all do just instinctively, the dream of like healthcare for a lot of people is to take a pill and be cured of like everything that could be wrong with you. And I think that that's the expectation for a lot of people. And I think that, there have been miracles like that with antibiotics and stuff like that. But with something that originates in your own body, I think it makes it harder. Mm-hmm. And what's interesting, as someone who has worked in a field of trying to expand access to healthcare to more people, like talking about earlier screening and like stuff becoming more accessible is really the fight to cure cancer. Like that is really like someone having a primary care doctor and not having to worry about whether they can make it to a primary care doctor or not is the cure for cancer. I mean, maybe it's a little bit of hyperbole, but I'm going to go with it. We kind of have the cure for a lot of these cancers, especially like a melanoma. The cure is finding it early and like having surgery or doing whatever needs to be done to get rid of it. And like, that is the cure. So it's, in some ways you can say that there's, this is a really sad conversation, but in other ways you could say it's actually really hopeful, because it's not really, technology or know-how or expertise that's preventing us from curing most cancers. It's people not finding it soon enough.
You are 100% correct. Like, if I think the field was being honest with itself, most of the reason that we have made significant improvements in cancer treatment is because we have much, much, much better screening and much better education. People kind of know, hey, if you have a suspicious mole, maybe you should get it checked. And it seems like second nature, but that's not by accident. That's by a very, very like specific and strong public health campaign, right? Or like, I should wear sunscreen, like public health campaign, you know what I mean? All of these things, like educating people about what to be wary of, what to look for, and, having people have access to a provider that could potentially give them a yes, no. I mean, these are all things that, and like, of course, like people delay treatment because they can't afford it or because they have, lack of access. And like, when you're talking about cancer, delaying treatment by even a matter of weeks or months, I mean, that can make or break sort of your survival. The other thing that we talk about a lot in the cancer field is the disparity among different ethnicities and survival rates. And I'm sure you can all guess who has the lower survival in the United States, right? Of course, it's people of color. And it's for a lot of different reasons, but most of the data shows that it's really due to lack of access and people's inability to afford care. So, and also, not to get on my soapbox here, but especially in melanoma presents quite differently on people who have darker complexions, because melanoma is a very dark tumor. It looks black, like on skin, but that color contrast is not as pronounced as somebody who is more darkly complected. So oftentimes dermatologists don't really know how to diagnose it, or they don't recognize it because it's not in textbooks, it's not taught, right? So you kind of have this like whole systemic problem that permeates into, all of these inequalities. So I agree. I mean, I think that healthcare equity, if we could kind of get even closer to healthcare equity, we would see massive declines in cancer, or I guess increases in survival, declines in late stage cancer diagnoses.
All right. And on that note, we are going to wrap up. Thank you, all of you, for joining us for this excellent discussion. And before we go, I'd like to give each of you the opportunity to promote any projects that you all have in the works, social media, etc. So Gloria, what do you have to promote?
Sure. Also, thank you again, Jamie. And also speaking with Teodora, this was great. So I actually write a blog. It is called Bare Bones Biology. It's barebonesbio.wordpress.com. I basically write about a topic in biology, kind of in the spirit of this podcast, where it's accessible to any listener, anybody at any level. So you can go ahead and check that out if you're interested. You can also follow. It's @_barebonesbio on Twitter. I also write periodically for Massive Science. I actually wrote about sort of inequality in the dermatology field recently. So you can check out my profile on Massive Science as well to read more of my work.
Thanks. How about you, Teodora?
So I write on my blog, curiouscortex.com. I write about neuroscience topics that just interest me and apparently other people. So you can find some of my writing there, and that has kind of got picked up by other people like Scientific American. And so if there's anything on Scientific American, I'll let you guys know, and it'll be linked there on curiouscortex.com. And then in the process of putting together I think it's going to be a nonprofit right now. We don't know what it is as of yet, but a team that I met at ComSciCon, we are putting together a mental health Google-like platform where people can just put in their questions, and we as the researchers go behind the scenes and get the answers in a very scientifically minded way. And mental health is very, very sort of, people that have mental health issues, they don't want to talk to anybody about it. So we thought this would be a great way to get that going. It's going to be called mentalliminded.com. We don't have a website yet, but it's coming. So that's exciting.
Great. I can't wait to check that out. And Jake?
Yeah, so I am, I've been working on a few things, but one of the things that comes to mind is the organization Jamie and I founded with some other friends with St. Louis Quarantine Support, where we were delivering food to people who were having to quarantine at the onset of the pandemic. We've kind of morphed into just kind of a hunger alleviation organization and a mutual aid organization. So my Twitter handle is @bankonjake. And if you're interested in how an organization like that would get going or something like that, feel free to reach out, and I can give you some pointers on how you could set up a similar mutual aid organization in your community. It's been really enriching experience. I think you can speak for myself and Jamie for doing that. So yeah, that's about it. I mean, and on Twitter, if you want to see me in my natural habitat, fighting with people about politics, you can find me there @bankonjake.
Great. Well, thank you all once again for joining us on our second ever episode of In Plain English. We have been talking about the paper called Tumor Microenvironments, Allies of Cancer Progression. You can find that free to download on inplainenglishpod.org. And once again, you can follow us on Twitter @PlainEnglishSci. That's PlainEnglishSci, or find us on Facebook at facebook.com/PlainEnglishSci. And you can find our podcasts on Google Podcasts, Spotify, SoundCloud, or wherever you listen to your podcasts. We'll see you next time for another paper presented in plain English.